to start the session, please. Can you hear me? Speak louder. Okay. I'd like to welcome everybody here to the Novartis Prize Award Ceremony. We're here to honour and congratulate 3 prize winners, giants in immunology who have more than 100 years of research between them. The prize has been awarded initially by Sandoz since 1990 and has been associated with the immunology congresses since 1992 and was changed in name to the Novartis Prize in 1995. Traditionally, there have been 2 prizes, one for basic immunology and one for clinical immunology. These prizes have often been shared by 2 or more recipients each. When the jury convened to consider the more than 50 nominations earlier this year, we rapidly found there were 3 very strong front runners. This presented a problem to fit 3 prize winners into 2 prizes, but one of the members, and I can't remember who, came up with the excellent idea of awarding 3 prizes, and this was approved by Novartis, so that is what we have done. We have awarded the Basic Immunology Prize to Ralph Steinman for his work on dendritic cells, the Clinical Immunology Prize to Hugh McDevitt for his work on immune response genes and autoimmune disease. And the Special Immunology Prize to Leonard Herzenberg for his work on this wonderful cell sorter that we all use. In each case, the prize work represents more than 30 years of sustained effort. That should not be a discouragement to younger scientists that they have to struggle for 30 years to get that far. But in this case, we recognise the fantastic contributions of these 3, which impinges on all our work every day. Lastly, I would like to acknowledge and thank Novartis for supporting and funding these prizes. These are the major prizes in immunology. They recognise the contributions of those who really change our field and affect the work we do every day. And so these prizes are wonderful recognition for those scientists. Now I would like to ask Jan de Vries from Novartis to say a few words and actually to present the awards.
So it is a great pleasure here in front of so many friends and colleagues, and on behalf of Mark Visman, the head of Novartis Research, and Thomas Adley, the head of Novartis Pharma, to hand over the Novartis Prizes for Immunology and Clinical Immunology 2004. The prizes are established to recognize groundbreaking individual achievements in the field of immunology. And as a sponsor of these prizes, Novartis honors innovative research in immunology, which is one of the challenging areas of modern medicine, but also one in which recent advances have led to the treatment of conditions once thought to be incurable. Immunology is a key area for medical research, and as new discoveries continue to provide therapeutic applications to many different diseases, which include autoimmune, inflammatory and infectious diseases, asthma, cancer therapy, and many others. Novartis' commitment to immunological research has resulted in the discovery and development of a number of successful drugs in past decades, most notably in the past cyclosporin, known under the brand names Sandinium and Nerol, which provided a breakthrough in organ transplantation. More recently, Novartis developed Elidel, a unique calcineurin inhibitor. The Novartis Prize for Clinical Immunology in 2004 goes to Hugh McDevitt from Stanford University for his discovery, mapping and characterization of major histocompatibility linked control of the immune response, and his observations that allelic polymorphisms in MHC proteins determine susceptibility to autoimmune diseases, which provided a roadmap for many investigators to probe the role of MHC proteins in susceptibility or resistance to autoimmune disease. May I invite you? And last but not least, Novartis awarded a Special Immunology Prize to Leonard Herzenberg from Stanford University of the first FACS sorter and the introduction of fluorescent-labelled antibodies as reliable FACS reagents, which opened the way for major contributions not only to the field of immunology, but to modern medicine in general. Your name is on it. Okay, Novartis congratulates the recipients with their achievements, and I'd like to give the floor to anyone.
Thank you, Jan. Now we will hear laudations and short lectures by each of the prize winners. So first I would like to ask David Sachs to introduce Ralph Steinman.
Dr. Hugh McDevitt, who was at that time an instructor at Harvard Medical School. So it's a particular honor to have him here tonight as well. I wonder if we're ever going to get these slides. I must say, with the modern technology, this happened to me very recently when I was presenting from my own computer. But I was told we're not allowed to have computers here, just a disk. When I had it from my computer, I would have just turned it around. What should we do?
This is not Swiss precision. Maybe we can change the order.
That was in on tomorrow.
No, not
All right, well, that's Ralph Steinman. Let's see, I think I can do it with this. Now, as we have been told by Irv Weissman, the dendritic cell is at the center of immune recognition. If that's true, then we can say that Ralph Steinman is at the center of immune recognition. Indeed, I searched recently on Medline for Ralph Steinman and the dendritic cell and came up with 244 references. But Ralph, while he is at the center of the dendritic cell in immunology, was not always interested in dendritic cells. Ralph was born in Sherbrooke, Quebec, about 100 miles from here, and grew up in Sherbrooke. He was born on January 14, 1943. He received his Bachelor of Science degree from McGill University with honors in 1963, and then went on to Harvard Medical School. You see him here on the first day of clinical examination in our second year of medical school. He's there on the right, you see. He was already thinking at that time about getting back into the laboratory. Now this is their graduation from Harvard Medical School. Ralph graduated magna cum laude in 1968, seen here with his parents and his grandmother. And you notice that there is another member of this group at the top of that list. It's another association of mine with Ralph Steinman. Now, as I mentioned a moment ago, we both graduated from medical school and went on to Mass General Hospital. He to an internship in medicine, and I to an internship in surgery. I recall my weekends on surgery, catching up on some very much needed sleep and studying for my next procedure, working very hard. Ralph recalls spending time during the residency at the nearby beach. You will notice, however, that he is carrying a copy of the New England Journal. In 1970, he moved to the Rockfeller University, where he maintained a similar style, as seen in this slide upon his arrival. He was a postdoc at the time under Xanthal Cohn. He and Xanthal Cohn published 35 papers together, among which is the landmark paper in 1973 describing the dendritic cells. Now, during this time,
I'm so grateful for this honor. I'm so excited that I have to read my lecture. And I deeply appreciate this celebration of dendritic cells. I feel that this occasion would be a good time to recall the early days of this field. It is for me a great honor and a very special personal pleasure to introduce Hugh McDevitt. Hugh McDevitt is Professor of Medicine and Chairman at Stanford University, where he spent the largest part of his career. Hugh McDevitt is known first for...
Jean-Francois, thank you very much for that, that wonderful introduction. For a minute there, I was afraid you were going to take over the major part of my talk, which is about TGAL. But I see that it didn't actually go into that. I also want to thank the members of the award panel for selecting me along with Ralph and Len to receive a really wonderful award, and as Andrew said, it has come to be the premier award in immunology. And I would also like to thank first Sandoz and then Novartis for continuing and establishing and giving a great deal of emphasis and support to immunology and to these awards as a way of recognizing immunology. Now, I should say that I came prepared with a CD for my talk in case something went wrong the way it did with David. And I didn't bring a written text, but I did bring a little note about what I'm going to talk about. And the story starts only about 6 or 8 weeks ago, when I was in the parking lot at Stanford, and I was rushing along. And a fifth-year graduate student said, "Hugh, where are you going?" And I said, "Well, I'm in a hurry to go to Israel." And he said, "Why do you want to go to Israel?" And I said, "Well, it's the 80th birthday of the man who invented TGAL." The student looked at me for a second, and he said, "What's TGAL?" Now, I'll bet, I can tell from the audience response, that that's the response of 90% of the people here. And that led me to make this a historical center of the review of the discovery of our genes. So today I'm going to tell you about TGAL, how it was made, who made it, what it
to the laudation for Leonard Herzenberg.
Last but not least, it is a pleasure and an honor to introduce the awardee for what is called the Special Prize in Immunology, Leonard Herzenberg. It suffices to walk around outside in the exhibition hall and peruse the posters to appreciate fully the impact of Herzenberg's contributions to immunology. There's scarcely a poster that is not adorned with a fax plot, and most of the equipment that you can purchase outside consists of cytofluorimeters in one shape or another. It is often, I think, with some disdain that intellectuals look at the development of technology, much more so even the reduction to practice of what is simply a good idea, but something that still needs the translation into a piece of equipment that works. And it is for this, I think, that we owe a huge debt of gratitude to Leonard Herzenberg and what he has accomplished. Now, I cannot tell you anecdotes about my share in any institution of higher learning with Leonard Herzenberg because he started graduate school the year before I was born. Nonetheless, for those of us who know Len well, whenever one visits a focal point of immunology, be it in Europe or in the US, it is more likely than not that one will find Len Herzenberg there making pronouncements that are certainly worth listening to. In 1952, when he started at Caltech for his thesis work, which was completed in 1955, he was exposed to the likes of George Beadle, Max Delbruck, Renato Del Becco, Sperry, Jim Watson, Barbara McClintock, and many others. It is hard to see how being exposed to these fantastic scientists would not have a shaping influence on the contributions of Leonard Herzenberg, and I think it has paid off handsomely. He claims, I think, and this is apocryphal, that the fly counter developed by Ed Lewis at Caltech planted the seeds for what turned into a cell sorter some 2 decades later, but I'm sure that Len in his speech will tell us more about the true origins of this machine. Having completed his PhD in 1955, Len, like so many other scientists of his generation, went to Europe for an intellectual shot in the arm and worked at the Pasteur Institute in the lab of Jacques Monod, immediately adjacent to Wolf and François Jacob. And it is here, I think, that we find many of the roots of Len's extensive connections with European immunology and France in particular. An interesting interlude, I think, is the event in 1957 when Len turned out to be drafted into the US Army. And as so many scientists of that generation, he had the opportunity of rendering service to his country not by donning a uniform, but rather working at the NIH. This, I think, is a part of immunological history that still needs to be written because a whole generation of physician scientists have entered the field precisely because of this opportunity. And it is most doubtful indeed that in the prevailing political climate south of the border, that exercise will be repeated. Be that as it may, at NIH, Len got interested in certain aspects of somatic cell genetics and had the good fortune, I think, of running into Leonard in Harry Eagle's lab around the end of the 50s. It is Leonard who recruited Len to Stanford, where he's been ever since in the Department of Genetics. Now, in Stanford, there starts an interesting episode in which genetics are combined with the production of antibodies. And some of the anti-MHC antibodies used to such great effect by others in the field were generated by Lee and Leonard Herzenberg. And it is around this time, I think, that Len became aware of particle physicists or engineers at the Los Alamos facility, and I'm sure
individuals who had constructed the prototype of the machine that Len at least recognized as being a cell sorter in the bone. And what happened then in the years 1969 to 1972 is that I've said the reduction to practice of what we now know as the FACS-1, the first truly operational cell sorter cytofluorimeter. I recall arriving in 1980 in the lab of Klaus Rajewski, where I was confronted with a FACS-2 equipped with the second laser, a monstrosity of a dye pump piece of equipment that spewed colored liquid all over the room in which the instrument was housed. A far cry from the neat boxes that we get when you look outside the exhibition. Now, I think the brilliance of this instrument cannot be seen detached from the application of monoclonal antibodies to cytofluorimetry. And here again, I think we owe a debt of gratitude to the Herzenbergs because unfettered by lawyers and their pesky materials transfer agreements, they early on agreed that it would be a good idea to share resources freely and actually make available antibodies and cell lines to those who requested it. Now that real money is involved, this has become a rare occurrence, but I think we should really go back to the late 70s and the example provided by Herzenberg. It's fair to say, and I think we'll be confronted with that in Len's presentation hereafter, that Len's social conscience is matched by his intellectual contributions. The prize is awarded, as the laudatio has already mentioned, for his contributions to developing and reducing to practice an instrument that all of us use, that has changed the face of basic immunology as well as clinical immunology as we know it. But as I've said, Len has remained active in a number of causes that seek to better the lives of many, not just scientists and not just those who would benefit from the fruits of our labors. It is therefore with great pleasure that I would like to introduce Len for his accomplishments and his fantastic contributions to immunology. Congratulations.
Thank you very much, Hilly, and thank you very much for the committee that chose me and for making this a 3-prize rather than a 2-prize thing so that I could be included. I always like that. I think it's important. I thought Leonard Herzenberg would be sitting in the front row so I could see her, but she's back there someplace. And she played a very important role, as I'd heard before from Hugh in his work, by making an important suggestion. And she played a very important role in my life and all my work. We've been married now 51 years, and we've been together all that time, and we've worked together, and this prize should be given to her one of these years, because she's really been in the beginning she wasn't a technician. In fact, she wasn't even a scientist. She was an English major, and we brought her into science and had many of our experiences together. And let me just start with the talk in the first slide. Just press this button. So there wasn't immunology before the facts, and I'd like to tell particularly the young people here a little bit about what occurred before the facts was put into the picture. And it was fluorescent microscopy, and in fact fluorescent microscopy was brought in by Al Kuhn, who was Hugh's mentor at Harvard. Well, I didn't push any buttons. Can we go back? Just leave it alone. I'll do it. Too fast. Okay, so fluorescent microscopy, you can read that, and the young students who are here should certainly read that and realize it. Lymphocyte populations, I have one of my former speakers said, Hugh said that Meadowar thought they weren't very important in the thymus, and perhaps the studies were done then, enriched populations separated by density gradient in general. And T and B cells were known. This was in the late 60s. Then there were events happening at Stanford. Remember, there was the Vietnam War. This was late 60s, was in the middle of the Vietnam War, and there was a lot of resistance. Stanford, I'm proud to say, was one of the places where there were protests, and you can meet Berkeley was involved as well. And that was when we were thinking about the cell sorter, and in fact, I got the opportunity to find engineers who didn't want to work anymore on war work at that point, and they were so happy to be able to work on something biological, medical. So Hewlett was one of those, and Bonner actually came in a slightly different route. And the first paper we published was in Science, as you see, in 1969. And there was a primitive machine at that point. It wasn't yet a fax. Fax came along, and we got lasers made available. And there were people at Stanford that were laser experts. And we got Dick Sweet, who was working for another company called Varian. Many of you have heard of it. And Bill Bonner. And let's see, was that Hewlett there? So, those are some of the engineers, and the first fax actually was built in our lab by these engineers. I spent, every day with them, telling them what had to be done, and wasn't satisfied if they could sort a few cells or 100 cells, and then they had to go into thousands of cells to make it worthwhile, so we could continue to do functional studies. And in 76, I think, Scientific American asked us by then was Leonard Herzenberg, and Dick Sweet. Dick Sweet is still active with BD. There's a sweet spot. If you get the area machine, you'll see that the droplets are being separated, and there's a sweet spot, which has two meanings: sweet, Richard Sweet, and sweet spot, which is sort of American kind of colloquialism. Okay, so if you look at this flow diagram, this instrument, this is back in the early 70s. Anybody who's been operating or using a current machine can recognize that it's still very much the same. It hasn't changed in 30 years until along came the area where the sorters use a flow cell rather than having the cells in the stream the cells in the stream and air interrogated by the lasers and detectors. So this was early machine. Then I went to Ralph lab, and why did I go there? I went there to learn a little bit of molecular biology and DNA sequencing and so on. That was coming up then. But before I went, I found that he published a paper on hybridomas in Nature in 75. I went there in 76 and set our lab. I think Barbara Osborne is here, and she was a graduate student at the time, and she and her husband, Dick Goldsby, started making T-cell hybridomas in our lab, and we went on with B-cell hybridomas. I can't give you the whole history in the time I've got available, because we're having a dinner, tonight. And so we had T-cell hybridomas, B-cell hybridomas, and did the first fax work in Milstein's lab. Actually not in Milstein's lab, but rather at Oxford. I had to go to Oxford, where Walter Bodmer had access to a fax machine. Milstein didn't think that fax was necessary until we left. And then fax became very important in the Milstein lab. So the reason for dwelling on monoclonal antibodies and the fax is I think they're really complementary tools for all of immunology and for many other fields as well. But this is an immunology conference, so we'll stick to that. if you look on displays, you see the droplets. These are droplets that are, frozen by strobe elimination coming down. And these are one of the original slides. I don't have pictures of my children from back then, but I do have pictures of our droplets. There's David Parks, who's still leader of the Vax development group in our lab. Been with us for 25 years. Exposal, who was one of the engineers I got from Lockheed about 30 years ago. And there's a picture, which I think is a nice one, which went into the Scientific American article, where David had developed with its help individual cell sorting, sorting individual cells into separate wells, so that you can do cloning, for example. This has always been viable cells. My effort always was to do it with cells that are alive, so you can do functional studies afterwards. Okay, so the first fact study showed, and you can read that rapidly, I think. Michael Julius, I've been told, is in the audience, and he's the president, he's now vice president of the University of Toronto. And since we're here, that's appropriate. That he found antigen binding cells, separated DMP binding cells, and showed that they gave rise to antibodies, antibody-producing cells making anti-DMP. Pat Jones, I don't think she's in the audience. She was involved in the reconciliation work, B cells, and so on. This is human cells. From the very beginning, looking at, since we had a machine that could work with cells in vitro, ex vivo, the leukemia classification was important, and finding fetal cells in the blood of pregnant women was one of the findings I thought was very important. Diana Bianchi was a medical student at the time. She's now a professor at Tufts, and she's still working on that. And so there's a connection between autoimmune diseases and why there are fetal cells in human blood. I don't have time to go into it, but it's worth looking into. It's a very active field right now, autoimmune diseases and fetal cells persisting in humans. Now that's from the California coast. You know what those two organisms, their organism is rather, seaweed. And under visible light, I think Randy Hardy may have taken this picture. Randy Hardy is here. In the UV light, you see that it has fluorescent molecules in it. These are where the glycolytic proteins come from. Glycorespiranol, phycocyanin, etc. So we were on the march then to increase the number of fluorescent colors that were available. We began only with fluorescein and rhodamine. And now we're up to 17 different colors. From jellyfish, not by us, but by some workers at Columbia, whose name I knew I'd forget, and I've forgotten them again. You all know it. GFP, green fluorescent protein, spontaneously fluorescent protein, and we selected some variants of it, so we could have not an extra color, but an extra marker. And a few years ago, we made this slide. David Parks says we should call high-dimensional facts, not multi-parameter, because he's a physicist. We got into this, and high-dimensional is correct. And this is obvious. You've got 3 lasers there, and you've got a lot of different fluorochromes. And you actually, anybody, including the students, maybe particularly the students, can get the facts to work with a number of colors. But as we go on, we'll see that there's Mario Roederer was a research, well, he was a postdoc first and then a research associate, and he stayed with me for 12 years before he finally was willing to accept a job that was better than his reality at the NIH in the Vaccine Research Center. And we named Roederer's Law after this kind of a slide. Moore's Law, having to do with computers, that the number of parameters increase every couple of years, and then the price goes this is only one side of it. Number of colors goes up every 9 years in this case. And you see we're up to Q dots. We haven't done that in our lab yet, but Mario, I share with me, he's done the Q dots. People with those are, but many of you know them. Okay, so high defects. The advantages, some of you, particularly the older members of the audience, they wonder, why do you want to have so many colors? Students, I think, immediately thought. So subset identification certainly is important, CD4, CD8, etc. Internal measurements, like for calcium, for DNA, mitochondrial indices, measuring kinases, phosphatases, and phosphoroproteins in general, caspases, and I should say, etc. High defects also. I'm sorry. We've got cytokines, chemokines, and redox. An interest of our Lee-Herzenberg and Leonard Herzenberg's interest these days is very much in redox because of AIDS, because glutathione is low in HIV, infected CD4 cells and CD8 cells. In fact, in all the cells of the body when you've got HIV infection. And you can measure inside cells by FACS, enzymes involving glutathione, biosynthesis, and then the oxidoreductases, thyroredoxin reductase, again studied in intact cells. So we can look for CD4, CD8 cells, but also any or all of these. And something I promised to say, I don't know how much time I have, I'll do it fast. EMA, ethidium monoazole, is very important in cytokine measurements. It's one bit of new information, maybe information people should be aware of or interested in cytokines. That you get false staining if you have any dead cells. Dead cells are almost always stained with antibodies to cytokines. We have weird cytokines which are not very abundant, like IL-4. It's important that you can stain the cells originally with EMA and then fix them and then permeabilize them, and then afterwards you can gate out the cells that were dead at the beginning. And EMA does that, and you can see that very clearly, I think, from reading the slide. EMA is available from molecular probes. I don't have anything to do with it, except science. The future of facts, we're still working on it in many ways, and companies are helping us a lot. Certainly, BD is improving detection, speeding up hardware. Lee-Herzenberg is working on increasing software power. Automation is an important matter, high throughput screening. And computers support is important. And high-speed sorting. Trade-off between the speed of cells going through and the signals that you get. And so problems to work with it. Problems in the far red, we don't have very many photons in the far red. And the future also involves that biochemical access to rare cells, stem cells, etc. Redox enzymes I mentioned. Signal transduction by phosphoprotein staining. That's something which Gary Nolan is in the audience, also a student of mine many years ago, right, Gary? At least 20 years ago. And his student, Omar Perez, who actually had time in our lab and has worked out phosphoprotein staining and signal transduction. And if you want to know anything more about the history and future of fluorescence, I actually wrote an article a couple of years ago for another prize, and it's available online and on our website. And the website's indicated books on rotary. So now, as he said, I've always been interested in social matters from the beginning. And I told you how the facts began with a lot of help from the Vietnam War. In fact, people didn't want to work on the war at that time. And another person who felt strongly about that was Jonathan Beckwith. And he wrote a book. I met him recently. I heard about him for many years. I met him occasionally. He's a very good molecular biologist. And making genes, making waves, of course, was the story of a social activist in science. He got an Eli Lilly Award, a prize, 30 years ago or so, and he gave his personal award to, I think, physician's responsibility for science or social responsibility for science, various organizations of that, and all his life has been active in social matters and been very important in that area, and gave me the extra push, I guess, maybe, to decide that I would give personal prize, important personal prize that I've been getting, to this very useful independent leader-supported website, truthout.org. It's the antidote to corporate news media. Lee is on the board, L.A.Herzenberg on the board of this, and I can go back to the early part of the history of Steve Weissman, who was at Berkeley and was involved in the Stanford demonstration I showed you at the beginning, is now an editor of this independent website. And there is another war going on, and I don't think many of us are wishing that continues any longer. So we've gone full circle back again. And that's it. Thank you very much.
Thank you. So I'd like just finally to thank all the speakers.
Hi, this is Rachel Gerstein. Tell me your best experiment on the facts.
Well, I won't tell you my best experiment, but I'll tell you a funny story where
Okay.
I had worked for months to make this construct, right? And I finally transfected it, and I thought I'd get expression, and I was going to, some GFP expression, so I'm going to go and check it out on the facts. And the cells start running, and I start seeing green, and I'm really excited. It's like my dream come true. The fire alarm goes off. Do you remember this?
Say what? Stop?
Fire alarm goes off.
Oh, so you had to leave the building.
So, I don't know, there's some kind of
And the
automatic
experiment was
thing.
going really well.
The experiment was going great. I was seeing the little green dots. So excited. And there was something automatic when there was a fire. The laser shut off. They had it. Yeah.
The chilled water probably shut
That was
off.
all. I was quite prepared. Yeah, I was quite prepared.
Then the laser shuts off.
Because that happened to me when I was on the fax scan.
No chilled water, no laser.
And the security guard.
I'm sorry, we got to pan over here to Kathy Seidel.
I was on the fax scan one night when there was a real fire in the building, a little one, a microwave or something. And I wouldn't believe my experiment. I figured, oh, it's going to be nothing. And this nasty-looking Matrix security guard came into this pink coat and screamed at me. And I, dropped everything and left.
Okay, back. So what'd you do? The laser shuts off.
Well, I wanted.
That was a little
I didn't
interlude
care
that Kathy
fire or
did.
not fire. I wanted to sit there and see myself, but because the laser shut off, and I thought, okay, this could be serious. So we had to all go outside and wait until everything was clear. But then I wrote down in my notebook, because I wanted to document what was going on. Experiment stopped. Fire.
Alright, cool.
It still works. Yeah.
Okay.
But I didn't really have all the data. I had to repeat it.
Let's move on to the other corner of the table, where we have Dave Tarlington and
I have a good
Anna
story. Another
Oh,
fax
hey,
story.
Kathy Seidel, your best fax experiment.
Well,
Bypassing the lay people.
It's
What's
not
that?
really.
Bypassing the lay people
John
here.
has a good fax experiment.
You don't count, buddy.
Well, no,
Okay.
my only fax story is that when I
Okay.
met Kathy before we got married, we started dating. I thought the fax really meant facsimile.
Like an
And she kept mentioning
FAX
this
fax?
FAX fax. That's all. She kept talking about working on the fax machine as if it was some big thing, and I couldn't understand because it's a fax machine. Like, what's the big
But
deal?
Judd, what was the experiment?
There was no experiment. It was just me being a
All
ding
right. Good job.
That's not.
Okay, Aaron can't. This is like a wedding video here. Okay, but I'm not the wedding singer. All right. So what was your best fax experiment or your best fax story?
I think your best fax story is your fainting.
Except you have to tell it, because all I remember is being on the floor, lying there. I was laughing at something or other.
It was one of these long sorts where we were there really late at night, sorting, I don't know, AL type and BL type, something.
It might have been one of those where Alan and I were doing 2-machine sorting.
Yes, I think you were doing 2-machine
sorting.
Sorting through the night.
You know,
That's right.
Gary Nolan got accused of doing a piss poor experiment for doing a sort on 2 different sorters. Okay.
That's right, to test the difference.
But you know what we did that was important? Is we figured out how to log out of the computer and continue sorting so that Len would not be billed. Because
that's how I sorted all my gene traps and enhancer traps. Okay, so don't tell me I knew that trick.
Yeah, we were laughing and goofing around.
We were joking around, and Aaron was walking around eating from a bag of potato chips, and I think I said something hysterically funny, can't remember what it was now, and Aaron started laughing really hysterically, and before you knew it, he fainted, because he didn't get enough air. He just couldn't stop laughing. But the sort went on.
The sort went on.
Yes.
Okay. Now I only got 14 minutes left on this tape. All right. So Dave Tarlington, what was your best fact story?
My best fact story, you've already mentioned it, was Gary being accused of doing a piss poor experiment. I remember laughing myself stupid about that.
Okay. All right. Okay. So that was Dave Tarlington.
Yeah.
Now hi. Hi. This is like a wedding video.
You're the official.
If you have a good fact story or any story, I'm going to take it. It doesn't have to be a fact story. This is Barbara Osborne, who I only met several months ago at Moffitt Cancer Center.
We did, yeah. I went and gave a seminar and found a Herzenberg lab.
Yeah. And you are Laurel Eckert, who's my next door neighbor Huayu's mentor.
I'm what?
You know Huayu?
Huayu?
Yeah.
Absolutely.
Her office is right next to mine at Moffitt, so we have a connection. Yeah, okay.
We are all related.
There we go. It's all together. And who are you? I don't know.
I'm with her.
Okay, good.
I just tag along.
That's all you needed to say.
Tag along.
All right. So, no fact stories. You want
Fact stories.
Because Len's address is on this tape, so I'm going to send him this tape. So I might as well burn up the last 14 minutes of it.
Okay, so my favorite fact story is working late at night in the fax room in Stanford, with lasers flying all over the place, and Len hanging over our shoulders saying, "What's the data? Give us the data. What's the data?" No, it was Lee, but sometimes it was Len. Len usually was falling asleep, but every now and then he was awake and wanted to know what the data were, and you couldn't even get the damn lasers.
Excited about the experiments. Think about it.
Because we're collecting the data. We're running the machine. We're collecting the data on the computer. We couldn't see the data, and they're asking us to interpret it before we even saw it. And they're interpreting.
We're not supposed to do that.
Hey, he's a PI. I can tell. All right.
He's vice president of Rockfeller.
Oh, okay. All hail to the chief, or sort of the chief. Okay. Yeah, sort of the chief. Paula? Paula, can I impose upon you for a fact story? You don't have to. Any anecdote will do.
Well, when I first wanted to come to the Herzenberg lab,
Yes.
Leah was giving a seminar at Wisconsin, and I went to her and I said, "Is there any room in your lab? I'd be interested in working." She said, "No, we have no room, but maybe you could work on one of the neighboring labs, come on,
Right.
sports side." So then I went back to my lab. The next day, I got a call from Len. He said, he asked me a few questions, and then he accepted me into the lab.
Okay, very good.
It turns out I was working with Bob DeMars, someone that he was friends and a great scientist. So he called Bob and he said, "What do you think?" And Bob gave me a great recommendation. So he took me to the lab, even though there was no room. And when we were there, literally there was no room.
So they made room for Paula.
They made room for Paula. I had a desk. There was no lab benches. I had a desk. I had a technician. She had a desk. We worked on our desks. And then we also used them though for earthquake protection, so that if the water started shaking, we could get under our desks. So they were not only a lab bench desk, but they were also a place to hide in case there was an earthquake.
Okay.
It was a great time.
Thanks. We're going to. This is Takeshi.
Yes.
Tell us your best fact story. Your best fact story. Or best Herzenberg man.
Big machine.
Big machine. That's it?
And smoking.
And smoking. Wait a minute. When was it smoking?
It's electric trouble.
It was what?
Electric trouble.
Electric trouble.
Right.
And the fax machine was on fire.
Right.
At Stanford.
Yes.
Did you start it on fire? Come on. Now you can own up to it now.
Another alternative is to have somebody, talk, one person to talk, the rest of us can hear, and then the next joke can be told.
Well, that's what I was going to say. If we have a general conversation, understanding Michael being the Montreal host, we should make Michael start by telling us the silliest thing that ever happened to him while he was in the lab. And he's got several of those. So
I have so many silly.
And then we can wander from
Only
there.
one, though.
How many different ways? Oh, we're going to do so. We're going to do a round robin.
One that he's not already told tonight.
Oh, but they don't know. No. And we can't remember the name of the players. It's really embarrassing. Well, looking for overlaps in the lab. I mean, they're all too young, Leonard. These people are.
That's true. We didn't actually overlap.
No, we didn't overlap
Yes.
with Mike.
Yeah, by a month or two.
Like 30 seconds.
Yeah, but we're not that. We're old.
No, but it would be nice actually. I think that some people said that they don't know everybody who's here. So it would be nice at least for people to go around once and say who they are as we talk. So just say who you are when you talk. He's Michael Julius.
Who are those two people down there? I want them off the table. Who invited you guys? Really?
The girl? Good. It looked good.
Okay, so I understand that plan. Madam Chair, could you control this tape?
She's speaking to you.
Don't you think that we should just bring out the domino?
The domino.
Lori, you said you left after he left. You got there after he left.
I came after he left.
Yes.
Yes.
I have no idea who you are.
And you still know domino stories?
Of course I
Well,
know
that's
dominoes.
No, this is Quebec.
Guys, can we get to the story? I only have 8 minutes on this tape.
We should do a nylon wool story.
Oh,
But he mentioned nylon wool.
See, we knew nylon.
But didn't he mention nylon wool?
He did.
Yes, he did.
He mentioned you.
Yeah, I heard.
Right.
I'm vice president of the world.
You were. I didn't know that you
No,
were
it's true.
vice president.
No, it's true.
All right, I'll make up for that gaffe. I thought he was vice president of the University of Toronto. It's only a part of the University of Toronto.
Now, so I'm diminished. Now I'm deflated. Sorry.
But there's another vice president here. In fact, probably at least 2 or 3. But the new vice president, at least it's new for me to find out, that Luke, Mike Young, Luke is the vice president
of what?
Rockfeller University.
Oh, really?
That's pretty high. Now, what's he vice president of? And I didn't get that quite clear.
What?
Mook of pain.
That's on tape, okay? Just so you know. All right.
Jesus, who's the president of pain?
Lori, she's not even there.
Right. I'm
That's
the
true.
president.
She is the president of pain.
Ooh.
Oh, you're in bad trouble.
Well, let's determine how many generations we have at the table. I guess I'm pretty close to the oldest here.
No, where's Henry?
Oh, where's Henry? Where's Henry?
I don't know. He was here.
He's so old he had to go to bed.
No, but Henry. When did Henry leave?
He left.
It's a good sign he can go to the bathroom.
So, Michael, when did you come and when did you leave?
69-70, but the story, the entrance story. So you have to know.
69-70, you were there
So
for
I
a
was,
year.
no, I was there from some 70 to 74 and a half, 75.
75.
Because we had a Sendai infection. And I was trying to finish up and I had to stay beyond, so I actually was a postdoctoral fellow for half a year. So I was the first BD fellow. I was the inaugural BD fellow. You have to know. Because nobody was unaffordable.
Did that go through our review board?
Certainly not. Certainly not. So I was headed for medical school. Actually, it just declined.
Not at the end, but at the beginning.
When it gets like this, you. And I had declined Edinburgh because they had an 8-year program and they didn't care that I had a bachelor's degree. And they were only going to give me a year off, so the object was, of course, to get away from home, and so I was going to go to the University of Toronto. I was accepted in the laboratory of Susumu Ono of evolution by gene duplication, City of Hope Medical Center, Duarte, California. So this is the summer, can you imagine, of '69. Can you imagine? So I go to the lab and he puts me on a this is a Herzenberg story. You don't know this yet. So he puts me on a project that involved a very large black flatulent chicken. Every time this chicken saw me, it passed gas. I can't actually really remember what I think I was trying to induce. I was trying to induce alcohol dehydrogenase. That's what I was trying to do with this chicken. So in parallel, unbeknownst to me, there was these geneticists up at Stanford who were trying to figure out immunoglobulin allotypes. So the breeding, because they had a Poschiavinus colony at City of Hope.
What's Poschiavinus? Wild mouse.
Wild mouse, and this is an important aspect of the story. Eventually, they were doing the breeding down at City of Hope, and the mice got sent. The blood. Thank you, Mike. Got sent from that laboratory up to this place north, where the Grateful Dead were. And I remember one afternoon I was watching them bleed animals. So they sort of had it pinned out and they took a big pair of scissors and they went click right here. And then some days later a telephone call would come back, putatively from him or her, that said, make 1329 with 1362. They were trying to map the immunoglobulin. Did you remember how I told you how they bled these animals?
Clip.
Right. So Susumu went, oh, we can't breed those animals, they're dead. So Len obviously had a conversation with Susumu and said, well, we're gonna have to. So do you guys know Susumu is still alive and he's back in Japan?
Japanese, yes.
Japanese.
But
Is he back in Japan?
Michael,
No, he did.
He
He got clipped.
So he was covering ass. The animals were quite dead. So he packs me up a little box of these, and what did I know? I knew about flatulent chickens. I had no idea about mice, especially wild mice. Can you imagine? This is my entrance into the Herzenberg
I
world.
never knew about the flat.
Well, I'm trying to embellish a little bit. So I'm with another red-hot pre-med, and eventually Susumu looks at me and he says, but you don't know Susumu. Literally, he was writing his book, Evolution by Gene Duplication, and he gave me chapters, and I said, he wants to know from me. So he said, you go to Stanford, you learn how to bleed these mice, so that eventually we can do our art. And he goes, if you lose one of them, don't come back. And remember, I know about flatulent chickens. So they put them in a box. We drive up to Stanford. There's a Grateful Dead conference up at Winterland, and I'm really I'm ready to go.
They're presenting papers.
We get a pass at the door. We're in Lee's office. Do everybody know Lee's office?
Yes.
The floor, the pillows, the hookah, you know Lee's office. Sorry.
At that time, it was a table. That was part of the problem.
No, stop. The table wasn't a problem. It was the bookshelves that were the problem. So, for reasons that are totally beyond me now, we decided to close the door. It's a miracle we decided to close the door. Have you ever dealt with wild mice? Have you ever?
Oh, they jump.
They jump.
Popcorn.
I have never seen a beast go straight up, so high. So we opened the box. The room was Susumu, if you lose one don't come back. And I was with this guy, and he didn't give a shit. He really didn't. And all I could do, and I remember grabbing one in midair, turned around, and sunk its incisors into my thumb, and I'm holding on the floor at it. So this was my intro to the Herzenberg. This is my intro. Wow.
By the Welsh. Whatever happened to that Welsh Foundation?
They get money out every year.
They turned into grapes.
Wasn't it in Austin?
No, it was in Houston. It was in Houston. I was in Austin, but the conference was in Houston. So I went to Houston.
Welsh Foundation gives out money for chemists every year, large amounts of money.
So they had a big conference, and this is how long ago it was. They were discussing at this conference about hypervariable regions, and maybe that had something to do with binding antigen. So I was fascinated by this conference because I knew very little immunology, and I was learning all this stuff, and then I, in my naivete, just felt I could just go walking up to this guy, and this guy had this long, he had that leather jacket with long fringe coming off of it.
Yeah.
You know?
Don't you still have it?
I was going, well,
When?
this is,
It's
you know,
framed.
this framed thing. So I went up to him and started talking to him, and